Good day, Biology 3 students. We're all the way into Chapter 5 with the structure and function of plasma membranes. We already did a lab dealing with the flow of molecules across a membrane, and now in lecture we're going to go into more details of that. We already saw in our macromolecules chapter that membranes are made out of phospholipids with polar heads. That is the part that faces either into the exterior of the cell or to the interior of the cell. And the fatty acid tails that face inward toward each other because they are hydrophobic. They are nonpolar and attracted to each other. In this image of the membrane, we see that underneath it we have the cytoskeleton composed of microfilaments. And we have a lot of these blue blobs that are the membrane proteins. Some of them are integral. They go all the way across the membrane. Some are peripheral. They are either in one side or the other, but they don't cross that lipid interior. Some of them form channels. Some of them are coils that go through. There's an enormous variety of these proteins, and they each have their own specific function. The model of the membrane as a whole is called the fluid mosaic model. Fluid is the phospholipid portion. Each phospholipid is not static. It doesn't always stay in that exact position. It can spin, they can flip, they can move as rafts around in the membrane, and that movement is limited by the mosaic of the individual proteins that are scattered throughout. And by weight, the membranes are about 50% protein versus 50% of the phospholipids. These little ring structures that we see on either side, that is the cholesterol. And because it's cholesterol, this would have to be an animal cell membrane. Cholesterol is only found in animal cells. The other thing that is showing up is glycoprotein. So glyco means sugar, so it's a protein with carbohydrates attached. And glycolipids, we have a lipid with carbohydrates attached. And these glycoproteins and glycolipids serve as markers on the cell surface and can have various functions, including identifying what kind of cell it is, identifying self, meaning that in my immune system, we each have these surface markers that indicate that it is my liver or my heart or my skin cells and not someone else's. And that becomes important in transplant technology. We see our phospholipid again. With its phosphate head, there is the glycerol, the 3-carbon sugar. There is the phosphate. Phosphates tend to donate their protons when they are in solution. That's what makes them polar. And we've got 2 fatty acid tails. One is saturated, so it's linear, meaning it has all single bonds between its carbons. That makes it very compact. And we also have one that is unsaturated. It has a single carbon-carbon double bond, so that would make it a mono unsaturated fat. And my video is covering up, but this says 1 or more double bonds between the carbons. And that, as we discussed in our macromolecules chapters, that makes it unsaturated because there is that bend, and that double bond means that we have fewer hydrogens around those 2 carbons. A saturated fat, each carbon is surrounded or saturated by hydrogens. In an unsaturated fat, that double bond means we have more electrons shared between the carbon atoms and not as many shared with hydrogen atoms. The bilayer of our phospholipids is because we have the 2 layers of those phospholipids. And if we put those in an aqueous solution, not actual membranes, but burst them open and have the phospholipids, If we put them into an aqueous or polar solution, they tend to arrange themselves with the polar heads facing outwards because the environment, that aqueous environment, is polar. It could be a bilayer, or it could be a single layer lipid sphere, and we can even create those bilayers in an aqueous solution. None of these are cells, but this way is how cells organize their bilayers. Those integral membrane proteins can take on very different forms. I've labeled, randomly choosing, the amino end as being external to the cell on this integral membrane protein and the carboxyl end being internal, but they could be reversed. It depends on the protein. And I showed that with this one that has multiple crossings of the membrane. I put the amino end interior and the carboxyl end exterior. And this one has several beta sheets. It forms a structure called a beta barrel, so it is hollow and makes a channel in the membrane. So we can have a single pass, multiple passes. We can even form a giant barrel across that membrane. And what I've drawn over here is just two phospholipids indicating that this yellow box is supposed to represent a cell membrane. And these transmembrane proteins, these integral membrane proteins, are made of protein, which means they have those peptide bonds, and it is their R groups that stick either into the fatty acids or away from the fatty acids that will determine how they are interacting with any other substances. This is an image of a cell membrane. Since we're looking at HIV binding to a CD4 receptor on T cells, this is a human cell. T cells are white blood cells, immune cells, and CD4 is cell determinant number 4. When researchers were studying the things on top of the cells, that was the 4th one they came across. And it is a protein. It does go across the cell membrane of the T cells. There is also CCR5. That stands for cysteine chemokine receptor 5. Basically, it is a multiply transmembrane protein. A chemokine is just a chemical receptor. All of our T cells have it. The HIV also sticks to this second marker, the CCR5, and that's what tricks our T cells into letting the HIV enter. This is a repeat of that previous image we had of the plasma membrane, but now it is showing here to show us that the exterior surface of the membrane is not identical to the interior surface. The exterior is where we have those surface glycolipids and glycoproteins that provide various types of identifiers on the cell surface, and the interior has to be able to interact properly with the environment inside the cell and anchor the plasma membrane to the cytoskeleton. So it's called asymmetrical. If we were able to chemically pull those two layers apart, they would not look the same on their surfaces. We're going to look at several different types of transport. This first one is diffusion. It's called passive transport because we don't have to provide any additional energy. The energy is just in the difference in the concentration gradient of a solute on either side of the membrane. So if we have the extracellular fluid has many of these particles and the intracellular cytoplasm fluid has fewer, as long as those molecules are small enough and nonpolar enough, They will cross that membrane freely until there is an equal concentration on either side. So as we start, the exterior is hypertonic to the interior, still hypertonic to the interior, and it will continue until we get an isotonic environment. Once we reach that isotonic environment, that doesn't mean that the movement of molecules stops. It just means there's no net movement. So if one goes from outside to inside, then one will go from inside to outside. In facilitated diffusion, we are still looking at passive transport. It relies on movements of solutes from an area of high concentration toward an area of low concentration, but those substances are either too large or too charged or too polar. There's something about them that does not allow for the free movement across a plasma membrane. In that case, we have some sort of protein channel, a transmembrane protein, and the interior of this channel will be hydrophilic, so that it can interact both with the exterior and the interior, and that will allow the molecules to flow from their hypertonic region toward their hypotonic region until it is isotonic across that membrane. There can be carrier proteins. Carrier proteins are different in that they change their shape as the molecules move. So in this case, we have a hypertonic environment. The channel is open to that hypertonic environment. When the molecules bind, the top closes off to that exterior environment and releases the particles to the interior. And we do this one at a time to be able to control the flow of those substances across the membrane. With osmosis, we are moving water. Diffusion is the movement of the solute from an area of high solute to low solute concentration. Osmosis is going to be moving water from a high water concentration to a low water concentration. And that means we're going to be moving from low solute, which is high water, to high solute, which is low water. So this is in a beaker with a semi-permeable membrane. And these particles are shown as being too large to move through the pores of that artificial membrane. So we can't just do diffusion to move the particles. They can't move. So in order to maintain a balance of proportion of water to solute, we can move water from where there's a lot more water per solute to the other side. The water rises, and now we have an equal proportion of water to each of those solute molecules. Demonstrating this with cells, these would be red blood cells. If we have red blood cells in an isotonic solution, we have an equal movement of water into or out of cells. If we put red blood cells in a hypertonic solution, it means exterior to the cell, there's high solute or a low proportion of water. The water will move from where there's proportionately higher water to where there's proportionately lower water to try to maintain a balance. Unfortunately, in cells without a wall, that causes them to shrivel up and probably die. If we place animal cells that have no wall in a hypotonic, that means the exterior environment has a high proportion of water compared to solute, and the water will rush into the cells, in this case, because there are no cell walls, burst those cells. Plant cells and other cells that have cell walls have some protection against this. If we place a cell in a hypertonic solution, just like with animal cells, the water will rush out. High proportion of water inside versus outside, and that will cause the plasma membrane to shrink. It's called plasmolysis. If we have a cell in isotonic, equal water in and out. If we place a cell-walled cell into a hypotonic, greater proportion of water outside versus inside, it will rush in, but plants also have this central vacuole that will prevent too much water from getting in, and the cell wall can prevent it from bursting. So we've probably all seen plants that have too little water. They start to shrivel. This is what my houseplants look like on a regular basis. I forget to water them, and that's called loss of turgor pressure, because turgor is the push of the water against the cell walls inside that cell. If we have adequate water, it would be nice and healthy. You could overwater a plant and that would dilute its nutrients, and that would also suffer. We saw a little paramecium in the past. It does have contractile vacuoles that help to pump out excess water, so it doesn't rely solely on osmosis and diffusion. We can have electrochemical gradients across a membrane. If we're going to move ions, charged elements, we have to have some sort of transport protein or channel. Ions or charged particles cannot freely move across a cell membrane. And then we can maintain an electrochemical gradient. It's chemical because these are chemical elements. Electro because they're charged, and that does impart a charge differential across our membranes. We can transport more than one thing at a time. Many transport proteins are designed to transport one thing in one direction. That is called uniport. So we're moving one thing. And whether it's one thing from outside to in or a different transporter that would be moving one thing from inside to out, still a uniporter. Symporter means we are transporting two things together and in the same direction, because sim means together. So A and B are both going from the outside in. A different symporter might have C and D going from the inside out. An antiporter is one protein that's being used to transport one substance from the outside in and another substance from the inside out. There's a lot going on this slide. This is an example of primary active transport. It's active because we have to provide energy. We have to provide energy because we are going to be moving particles against their concentration gradient. So this is the sodium-potassium pump. It's a pump because we're having to use energy to direct each of these ions against their concentration gradient. We maintain this in our cells so that our extracellular, outside the cell space, remains high in sodium and low in potassium. And the interior of the cell is maintained with high potassium and low sodium. We accomplish this with this energy source called ATP, adenosine triphosphate. It is a carbon-nitrogen ring structure that has 3 phosphate functional groups on it. And the hydrolysis, the break-off of individual phosphates, releases more energy than is required to break that bond, and that released energy is used to do work in our cells. So to maintain this differential of our sodium and potassium ions, this transport protein will first bind to 3 sodiums that are on the inside where sodium is low. ATP energy forces a conformational change, a change in the shape of that transport protein, closes it to the interior, opens it to the outside, and now we're maintaining a higher concentration of the sodium ions. Once that is complete, 2 potassium ions bind, and we release the ADP, adenosine diphosphate, and that causes the conformational change to close that channel to the outside and open it to the inside. And now the channel is restored in function to go back and receive 3 more sodiums. This is called primary active transport because we are directly using that energy from ATP to move sodium ions out and to bring potassium ions into our cell and constantly maintaining this unequal distribution across the membrane of both of those ions. This is secondary active transport. We're going to use the fact that we maintain high sodium on the outside and high potassium on the inside to move glucose across the membrane. So it is symport because 2 things are moving in the same direction. We've got glucose represented by these little gold bars. We've got our sodium is still being represented by these little hexagons. And now, instead of just a static channel that closes off on one side or the other, we have this sort of rotating protein that is transmembrane. Because we have this high concentration of sodium, the natural inclination would be for facilitated diffusion to allow sodium to pass through the membrane toward the interior where sodium is low. We have high sodium outside, we have low glucose. But in our cells, we need glucose as an energy molecule. Even if it's in low concentration outside the cell, we need to bring that inside the cell to use in metabolism. So sodium could, if available, use facilitated diffusion to go through. Glucose would have no inclination to move into the cell because we have a low concentration outside and high inside. But we use this secondary active transport, the fact that we've already used energy to keep high sodium outside the cell to now provide energy for active transport of the glucose. 2 sodiums and 1 glucose bind to this transporter that then rotates and pushes the 2 sodium inside and releases the glucose. The sodium-potassium pump would then take 3 sodium and push them back out, maintaining a constant level of that high sodium to keep bringing in our low glucose. The thing that prevents this high concentration of glucose from going back out by facilitated diffusion is that as soon as glucose is brought in, it gets phosphorylated. A phosphate group gets attached. And then glucose with a phosphate attached is not the same as glucose. And that maintains the low glucose outside and the higher glucose immediately converted to glucose phosphate inside. Other ways of transporting material across the membrane is phagocytosis. Cytosis means a cell process, and PHAGO means eating. So it's an eating cell process. So the plasma membrane literally engulfs some particle and brings it in. That vacuole can fuse with a lysosome, the eukaryotic organelle that contains hydrolytic enzymes that carry out hydrolysis or breakdown reactions, to digest this. and that might actually be a food particle. It could be bacteria. It could be pretty much anything that the cell can engulf and break it down and extract the nutrients from it. Pinocytosis is for bringing in water. So we can certainly move water by osmosis, but if we need a larger volume of water, we can just pinch off the membrane and bring in that water. So we need more water than osmosis alone can bring into the cell. Pinocytosis can do it. Receptor-mediated endocytosis. So we need some sort of receptor, a specific interaction. It's going to use a cell process to bring in matter. So it's targeted to a single type of substance. There'll be some sort of receptor on the plasma membrane. A ligand is anything that is binding specifically to a receptor. And then the interior of the cell membrane has this protein called clathrin. When the ligand specifically binds to its receptor, that triggers the endocytosis so that we have brought in the desired material, and now we have this vesicle that is coated with the clathrin. And that is a signal to the cell that this specific molecule has been brought in, and those molecules can be used for the purposes that the cell needs. Exocytosis is the opposite of endocytosis. Endocytosis brings matter in. Exocytosis moves stuff out. So we have the vesicle that has waste materials or some protein needed by a nearby cell, or it just needs to be excreted. The membrane of the vesicle fuses with the plasma membrane and the contents get released. Whirlwind tour of membranes. I have the lab up already. I'm going to be spending all day Monday grading your previous labs and getting caught up so that everyone knows where they stand in the class. On Wednesday, I will be getting the study guide for exam 1 put up so that you know exactly what to be expecting and where to focus your study guide.